To understand the function of neuronal circuits, it's very useful to have a so-called wiring diagram of how each neuron in the circuit connects to each other neuron at the level of the individual synapses between them. In order to get this kind of data, you can record with electrodes from pairs or handfuls of neurons, or you can optically image and infer connectivity from functional data. But a direct method is simply to look at the underlying anatomy. What we did is image the entire brain of an adult fruit fly using an electron microscope to see these connections between neurons at incredibly high resolution. The problem is that this anatomy is at the scale of the wires in a computer chip. 100 nanometer diameter connections between the neurons, or even finer, are common. But the fly brain is almost a millimeter in extent. This means we had to take many, many pictures and assemble them into a coherent volume so that we could manually reconstruct the connections between neurons and circuits of interest to us. The full data set of this fly brain comprised 21 million images and 106 terabytes of raw data on disk. To meet this scale challenge, we assembled a large collaborative team who brought skills from diverse areas spanning tissue processing and sectioning, mechanical engineering, software engineering, and large-scale image processing. Together, we developed a custom tissue processing, sectioning, image acquisition, volume assembly, and neuronal reconstruction pipeline. We improved our sample preparation methods to maximize both high membrane contrast and quality of ultrastructure preservation. We sliced the brain into about 7,000 40 nanometer thin serial sections and imaged each section using custom engineered high throughput transmission electron microscope systems. We wrote new software to stitch and align the data set into a coherent image volume. Finally, a team of neurobiologists traced neurons and circuits of interest within the data set. We focused our efforts on the fly's olfactory system because the anatomy of olfactory neurons has been very well characterized at the light level. This meant we knew what to expect when reconstructing these neurons using the electron microscopy data set.
We reconstructed all the olfactory neurons, providing input to the mushroom body, a part of the fly brain used for associated memory formation and recall. This complete survey resulted in not only the expected set of olfactory projection neurons, but also new and unexpected circuit elements and connections. For a variety of cell types, our electron microscopy trace neurons recapitulate the morphology and projection patterns of known neurons imaged at the light level. We developed a bioinformatic pipeline to merge electron microscopy reconstructed neurons and neurons from light microscopy morphological databases. This enabled co-visualizations and geometric analysis of these neurons, as well as rapid cell type identifications of neurons traced in the whole brain volume. In order to identify the cell types of reconstructed neurons, we took advantage of the stereotypy of neurons across different fly brains.
The principal neurons of the mushroom body are called kinine cells, which receive dendritic inputs from projection neurons. Most of these inputs are thought to be olfactory, but it was not previously possible to map them all. In addition to their inputs, kinine cell dendrites also make output synapses, but their postsynaptic partners were unknown. We took advantage of the synaptic resolution in this whole brain volume to identify the partners up and downstream of kinine cell dendrites. As expected, olfactory projection neurons provide the majority of inputs to kinine cells, but an unexpected new cell type was also discovered to provide inputs that are likely multimodal. Additionally, we discovered that kinine cell dendrites preferentially target only a small subset of available cell types. While we focused on the mushroom body, there are over 20 laboratories from around the world tracing their circuits of interest in the volume. Their work is already spanning the entire fly brain and advancing our knowledge of connectivity within and across brain regions. We have also established internship programs with several universities over the years, creating opportunities for students to gain hands-on experience in research environments by working with our scientists to reconstruct neural circuits in many different areas of the brain. Additionally, we are providing a pre-configured tracing environment that includes the cat-made tracing software, the neurons presented in this manuscript, as well as scripts to download the entire EM volume. Scientists can trace any areas of interest to them and use import-export functions built into the cat-made software to share traced neurons across laboratories.